Good afternoon, everyone. We want to welcome you to the 145th session of the Congressional Biomedical Research Caucus. You know, this year we put briefings on about cancer, Alzheimer's, HIV. We did avian flu, and as I've always said to you before, we from the Joint Steering Committee of Public Policy would always welcome any topics that any of your offices might like to hear on biomedical research. Our chairs have worked hard in the past to achieve funding for the NIH. Sherry Bollert, Mike Castle, Lois Capps, Rush Holt, and of course Congressman Joel Heffley from Colorado, who is present here today, and we're delighted to have him. These men and women have worked hard, and you know, in the last several years we were able to double funding for the NIH. But unfortunately, in the past years, because of massive fiscal constraints, and it's a bipartisan effort. This year, the funding in the House has absolutely been flat funding the NIH for the first time really in nearly 40 years. In the Senate, Senators Specter and Harkin were able to put $200 or $300 million more in the NIH bill. We want all of you in all of your offices to look carefully this fall. You know the NIH needs the momentum to keep on going forward, keep the United States globally competitive, to reduce medical costs, and also to provide jobs for Americans, never mind the factor of health and long lives. So it's vital, and I hope that in all of your offices, you will watch out this fall when the NIH comes up in conference and in the final bills that close out this year, that you'll be able to achieve some increase for the NIH. Today we're going to learn about strategies for treating patients with genetic disorders before birth using hematopoietic stem cells. In particular, also about chimeras, what they are, how human-animal chimeras generated by the use of human stem cells can serve to facilitate scientific advances in human stem cell biology. We're privileged to have with us today a renowned world scientist in the field of treating patients with genetic disorders. Dr. Esmail Zanjani is a professor of medicine at the University of Nevada. He received his BA in biology and his MS and PhD in experimental hematology from the New York University. Dr. Zanjani has been a leader in the development of large animal models of in utero stem cell therapy and scientific advances in human stem cell biology. It's really a pleasure to welcome him here today. Dr. Zanjani.
Good afternoon. I'm actually also chair of the Department of Animal Biotechnology at University of Nevada in Reno. And that gets me into use of animals for the studies that I'm going to describe. My main area of research is assessing the ability of human stem cells to work in vivo, what they can do, not just what they can do in culture, but once you put it in a body, what they could do in there. And this is an extension of what was just mentioned of my long-term interest in using stem cells and gene therapy in treating patients before birth. Now in our studies, we inject, or better word would be transplant, human stem cells into animals. As a result, we develop human-animal chimeras. So I thought, and I was asked to talk about what chimeras are and what useful purposes they could serve. And I thought I'd begin by describing what the Greeks thought a chimera is. When the slide comes on, it is an awesome creature, fire-breathing, with a head of a lion, body of a goat, and the tail of a serpent. Actually, legend has it that the hero Bellerophon, riding the winged horse Pegasus, slays this creature. It's interesting, the horse is a chimera too in this particular case. But as the next slide shows up, we will learn that the scientific definition of a chimera is much simpler than that. It basically involves that human and animal chimeras have to have at least two different genetic cell types within their system. And to be considered a chimera, both of them have to be expressed. Now, there are a lot of naturally occurring chimeras in humans and animals in nature. And these generally involve blood cells. And they're at very low levels, so that they're really microchimerisms. And the best example of this is the fetal-maternal microchimerism, which results from the transfer of some blood cells from the fetus during pregnancy into the mother. And the net result is that these cells sometimes can last for years in the mother, and sometimes they could cause problems. There have been certain cases of cancers and things that have been attributed to the fetal cells that had entered the mother. Now, you know that generally chimeras, especially human-animal chimeras, raise a number of ethical and other concerns. But it turns out that human-animal chimeras that are being developed by the use of human stem cells in animals are proving to be very useful, extremely useful in both scientific and as well as clinical arenas. And that's what I'm really going to talk to you about. There are a variety of ways an animal-human chimera could be of use. For example, on a scientific basis, we could put the human stem cells in these animals and follow where they go. And this will tell us what particular stem cell type may go to a particular organ, and so that we can identify and isolate organ-specific human stem cells. This could be very useful in the regeneration type of studies that are being done, or if you want to regenerate an organ for transplant later on, perhaps using in vitro or in vivo technologies in the humans directly. The other way is that these human-animal chimeras become extremely useful in generating both generic as well as patient-specific organs and cells for transplant purposes. Again, when the slides show up, I will give you an example of this. Here's a scenario that you could follow, for example. A patient walks in, needs a liver for transplant, and the patient is appropriately put on a waiting list. Now, at this point, it is possible to have had generic livers that could be used to transplant this patient almost immediately. Or the patient wishes to have an organ, a liver, generated specifically to his or her stem cells, in which case you will ask to take about 1 or 2 ounces of the patient's bone marrow, which is done as an outpatient type of procedure. You will then take stem cells from these bone marrows, transplant them into fetal sheep or pigs. These are the animals we work with. And in about 1 to 2 months, you will have livers in these fetuses that you have transplanted. And you can transplant 6 or 7 fetuses with that 1 ounce of bone marrow. You will have livers that would have between 10 and sometimes 20 or 30% human liver cells. Now, of course, is specifically from the patient's own stem cells. In other words, it's autologous to the patient. Now, you can transplant either a lobe of that liver or the whole liver as an auxiliary liver to the patient. Now, the patient's body is going to reject the animal part of it. So about 60, 70, 80% of the liver that you have transplanted will be rejected. But the part that is patient's own will be accepted, and liver has a very nice ability to regenerate. So in short period of time, you're going to have a liver that's functional. And I'll show you that in the data. Now I forgot what my next slide was going to be. There are a number of ways of generating human-animal chimeras. Now, it's possible to inject human stem cells into an animal at blastocyst stage. This is at the embryonic development stage. And that's where the problem, of course, lies. If you were to do that at the embryonic stage, the majority of the animal's body will be human. This may include the central nervous system and some of the other organs. This may or may not be compatible with the life of the animal. And therefore, we avoid that. You can also inject them at a time when the animal is a fetus. And if you choose the time right, when the fetus has not as yet developed the know-how immunologically to reject a foreign cell, such as human cell, you're putting it in a sheep, for example, it will then accept it as such and allow this to engraft and do the kinds of things that I'm going to talk about. You could also inject the cells when the animal is an adult. In the adult case, there are problems because the immune system tends to reject these cells, and unless you bring immunological suppression to these animals, you're not going to really use.
We'll be back.
You'll be back, okay. That's all right, no problem. Would my stick work?
Your laser, yeah.
No, I mean, I have no.
Oh, do you have another computer?
No, I don't.
Oh.
What I was going to show you, actually, the next slide, was going to be a 3-year-old sheep that was human-animal chimera. 3 months before this animal was born, it was injected with human bone marrow stem cells at about 55 days of gestation. Now, sheep have a gestation period of about 145 days. But the nice part about 55 days is that they're immunologically naive. And so you put the human cells into them, they think it's their own, and these cells get distributed widely throughout the body of these animals to various tissues and organs in the system. And after they're born, there is no side effect to these animals. They look fine. I had pictures to show you, rather nice ones too. And you will see that if you were to examine these animals after birth, even at the 3 years of age, you will find that nearly all the organs you look at, blood, liver, pancreas, lung, skin, they all have components that are human. Now in the liver, where we spend a lot of time on, it turns out not only the liver cells are human, but also structurally, the blood vessels, the structures that make up a liver are also human-derived. We are creating, actually, well, I don't want to use the word creating, we are generating or developing a functionally complete unit. And we know they're functional because we can detect human liver proteins, such as human albumin, in the serum of these animals for years. We have followed some of these animals for now about 5-6 years. And the level of human protein activity is fairly high. If you look at the pancreas of these animals, you will note that they are making human islet cells that make human insulin. And these also persist for a number of years after the transplant. We've done this with human adult stem cells, and in one particular case we've used hematopoietic stem cells derived from human embryonic stem cells, one of the approved lines, and transplanted, and we found that the pancreas of this animal is also making islet cells that make human insulin. The mechanism is very important. How does a human stem cell get to change itself into, or give rise to, liver cells that are human and functional, or pancreatic cells that make human insulin? Now there are two possibilities. One is a mechanism called fusion, where the human stem cell fuses with the liver cell, for example, of the animal, and confers on it some human characteristics. Now, fusion would be a problematic thing, because if an organ was generated using the fusion mechanism, every cell in that organ would have an animal component to it. And if you were to transplant those cells back into the patient's body would reject the entire thing. It can't just accept part of the cell as being human. The other part is animal and it's want to reject it, and so you got a damaged system. Now, in mice, in some other species where human stem cell transplants have taken place, fusion appears to be a major mechanism. So we were looking for a system that did not offer fusion as a primary system. And it turns out this fetal system that I've just described does not use fusion as a mechanism. Here, a change occurs in those stem cells that find their way to the liver, and there a differentiation, a process called transdifferentiation takes place. We have not yet seen a single cell in the liver or pancreas of these animals that are product of fusion. And I think that's really very important. Now, I think my talk is going to be awfully short. There are other considerations when you deal with an animal-human chimera. There are ethical issues obviously raised. I'm not an ethicist and I'm not going to address that, but there are other possibilities as well. For example, the use of, maybe I could, okay, we'll skip some of these rather quickly. Here was the definition, the scientific definition. And I just wanted to bring up one thing that I hadn't mentioned, and that although the naturally occurring chimeras are microchimerisms, chimeras that are created by organ or bone marrow transplants could have a higher level of chimerism. For example, in bone marrow transplants, you could have as much as 90% of the blood of the patient to be of donor type. So you will have a higher level of chimerism. And these were the uses of what an animal human chimera could be used for. And this is where we were concentrating on to generate generic or patient-specific organs for transplantation. And this was the scenario I was following when a patient walks in and needs a liver for transplant, and you can either provide them with a generic one for an immediate transplant, or they can create a patient-specific organ for transplant purposes, as I described it. And this is, of course, based on the reality that there is a great need for organ transplants. There are a number of patients waiting for these organs to be transplanted, and we know that a large number of these patients die because we just don't have enough donors for that purpose. So there's a great need for cells and organs for transplantation. And of course the current sources are cadavers and live donors, which is not adequate. Possible solutions are xenotransplantation using animal organs. You know, xenotransplants have been done. There are a lot of chimeric individuals with parts of pig valves, heart valves in their system, or cows. And some organ transplants have also taken place. The other, of course, and here's the promise of it, is the stem cells or organogenesis, where you can take fetal or embryonic organs and develop them into a larger organ for transplant purposes later on. And of course, this is where the humanized cells or organs generated in animals by human stem cells is what I'm talking about here. This raises the potential that you would have an unlimited supply of cells and organs, and of course availability on demand. I mean, it's literally possible to have a farm where you have large numbers of these animals transplanted with stem cells with different genetic makeups, and then that could match up with a particular patient. And I mentioned how you could, at what stages these could be generated. Now here's the sheep at 3 years of gestation. I want to show you these pictures. And 3 months before its birth, It was injected with human stem cells from human adult bone marrow. Or you can do this, of course, under ultrasound guidance, but I thought visually this shows where the injection takes place. And these animals are born healthy and there's absolutely no side effects at all. I mean, sometimes infections occur, but it's been very rare. And as I said, if you were to examine this animal, you will find that its lung has about 2 to 8% human cell activity, liver as much as 20–30%, muscles, guts have variable levels, blood as well, heart, pancreas. The central nervous system, the way we inject them, which is interperitoneally, it's the space between the body wall and the gut, doesn't get itself too readily to the central nervous system, so the levels are very low. This is the same information, except to show that in liver, pancreas, and blood, where we have really spent a lot of our time, these cells are functional, human functional cells in the system. Now here's the liver data. Here's an animal liver and here's a human liver probed with this antibody that makes the human components brown. And here is a chimeric sheep liver that was made chimeric with human cord blood stem cells. And you can see a significant portion of the cells in the system are brown and therefore human. And these are the other structures, such as the biliary ducts and blood vessels that are also human. The brown version tells us that. And therefore, they do actually function to produce human protein that can be detected in the circulation of these animals for years. And I talked about the fusion and the transdifferentiation mechanisms. Here is the data supporting that fusion is not an important mechanism here. Here is human liver probed with a green probe, and here with a sheep liver with a red, and here's the chimeric liver. You can only see red or green. If you saw the two together, you would know that there's fusion, and we haven't seen even a single one yet. And as I mentioned, the transplantation of humanized organs containing fused cells will result in vigorous rejection of fused cells. And the other thing is, these fused cells seem to facilitate transfer of viruses into the recipients. And so it's a bad idea to use these fused cells for transplant purposes. Here's the data on the insulin-producing fetal sheep pancreas. Here is the sheep liver, and again, this is sheep pancreas, and here a human pancreas that's been stained with human antibody to human insulin. Again, the brown is the indicative force, and here you can see a liver from a sheep that was made chimeric with human cord blood stem cells, and these islet cells are making human insulin. And here is one where human insulin is being made by islet cells developed by hematopoietic stem cells derived from human embryonic stem cells. So what are the uses of some of these human-animal chimeras? One can easily use it to study the in vivo activity of human stem cells. You can generate genetic cells and organs, as I described to you, or you can generate patient-specific humanized cells or organs. The other issues that I think come up are ethical, some religious, moral, scientific, but this is something that society has to deal with. But there are safety issues and institutional oversights that need to be looked at. One of the ethical issues, not dealing necessarily with some of the religious and other ones that I listed, is our willingness to sacrifice non-human animals for human needs. Is it based on something other than just merely our ability to do it? Just because we can do it, should we do it? It's something that got to be looked at. Now, do our use of animals for food, clothing, etc. provide justification using these animals for the purposes that I've just described? Should experts make all the decisions, or should there be a broader general citizen impact? Safety issues, I only can come up with two. Well, actually, I come up with three, and I'll cover the third one in a minute. One is infectious agents. We've been using farm animals, the kind we work with, for millennia to sustain us. Now, have you gotten used to some of the agents that could create problems for us? We probably have, but this is something to be looked at, I think. Nevertheless, funding through NIH would really serve to help us with these kinds of activity. And the problem gets a little more complicated if you use fused cells for transplant. The other safety issue is you're developing these human-animal chimeras. Are these going to be transferred into offspring of these animals? Any human characteristics going to go into new lambs or piglets that are going to be born? Well, it turns out that while there is wide distribution of stem cells to all tissues and organs, once you transplant the human stem cells into these animals, they don't go to the reproductive system. Germline cells are not involved. And the result is that we've done this on repeated occasions, where we've bred these sheep to see what the offsprings show, and there is no human cell activity in these offsprings. I think that's really, perhaps in safety issues, even more important than some of the others we talked about. The institutional review boards at universities and other research institutions have been very effective in providing ethical and biomedical guidelines. They're really very effective. They prevent you from doing stupid things in animal and human studies that we tend sometimes to want to do. So I think a careful oversight by relevant IRBs should take care of this and actually facilitate the kind of research I'm talking about. So here's that scenario again, but now I have added other organs to it too. It doesn't have to be limited to liver. Now, liver is a very interesting organ because it regenerates, and I think it'll be probably the very first organ to be tried on. But there are others that one can use as well. And so and this is the last slide, and the fetal approach really tells us. There is widespread distribution of stem cells in these animals, which results in the generation of multiple humanized organs. One source of stem cells can make you all kinds of organs in the system. The induction of tolerance, because you put these cells in so early before they're immune tolerant, they become tolerant to that specific source and actually allow these organs to persist in these animals for years. So you don't have a window of opportunity. You have actually years of opportunity to use these cells or organs. The humanized organs appear to be generated without fusion, and I think that's very important. The central nervous system so far is minimally affected and takes away that yuck factor, that you're talking about human consciousness being trapped in an animal's body. Let me out type thing. And the reproductive systems are not affected, and I think that's just as important in this particular issue. And that sort of summarizes what we do, and I think that the human-animal chimeras should really be supported because, and funding from NIH is very important in this, and I hope you guys help with that.
Thank you very much, Dr. Janzani. And now we will entertain questions for the good doctor. So tell us again, some grown man needs a liver. You take the stem cells from him, put them in the womb of a young lamb,
Yes,
and then grow the liver for him?
that's correct.
Well, Esmail, has it been done yet? Have you achieved this yet?
It hasn't. Well, we have developed livers that are specific to individuals, including myself. I've given stem cells from my marrow, and there's a bunch of sheep that have livers that are specific to me. We have tested some of these liver cells in vitro by taking a few of the liver cells that are human, and my lymphocytes, my body doesn't appear to reject them. So the idea that your body will not reject it is there. It has not yet been put in human subjects, but we've done proof of principle studies where we have done the same study between pigs and sheep, where we have transplanted pig stem cells into sheep and vice versa, and taken the livers a month or so later and transplanted them into the other animal. As we predicted, the animal's body rejected the other animal's cells but kept its own, and those regenerated into fully functional liver, where we've then gone in and taken the animal's own liver out, and the animal survived for months and months afterwards.
And are you absolutely certain that this is not transmitted from the donor of origin, so that a human stem cells would then stay in an animal and then be transmitted by reproduction to another animal, so we'd have a bunch of animals that were partly human?
We are as certain as we can be. We have bred 19 different chimeric animals to multiple male or female, as the case might have been. And in neither of these, examining some 63 lambs, has there been ever an indication of human cells in the offspring of these animals. I think that's not, it's very, very difficult to affect germline cells by these kind of transfers.
Yes, sir.
I was going to say that one way of addressing that issue is simply to sterilize the animals, so that then there's no possibility of being transmitted to the germline.
That's a possibility, although we feel that that adds a problem that may kill some of our fetuses. But even with gene therapy, you can't affect germline cells that readily. I'm sorry.
Yes.
That,
I'll take it.
okay, I'm
Yes.
sorry.
I actually had two questions. The first was, I was struck by the fact that you're actually getting transdifferentiation from an adult donor. And I know that that's been quite contentious in the scientific literature, and so I wanted to know if you could address that. And the other question is, if you had this image of using embryonic stem cells, you sort of alluded to this, to create a bank of typed people, so that, we normally now type people for transplantation, and so rather than having it being autologous, whether you sort of envision a bank making system.
Yeah, that's where the term generic came from. We feel that we can take the most common type of HLA types of humans and basically generate a whole bunch of animals that have these organs for that purpose. We think we could do that. As far as transdifferentiation is concerned, we get much more human cell activity in these organs when we deal with cord blood stem cells as opposed to adult. But we do nevertheless get what we are calling a transdifferentiation. It may very well be that, we know that even the highly purified stem cell populations are heterogeneous in nature, and they may actually not be the same stem cell that makes blood cells that are developing these liver cells. So we think differentiation occurs, but transdifferentiation, that's the term we use.
Just a moment for the mic, please.
I was wondering about, you're alluding sort of to the possibility of of not just using this for liver transplants, but for other organs in the future. But then when, with the numbers on it, you had fairly low percentages of human cells. And for an organ like a heart, that would seem to be fairly problematic. If it rejects 97% of the heart, then it's not going to regenerate. So I'm wondering, do you think this, you alluded to having hope for other organs beyond the liver. Just sort of, if you could speak more, if you hope to increase those percentages somehow, or what's the...
Yes. We can use for heart. I mean, heart is going to be an extremely difficult kind of thing to work with. But we can increase the level in the heart to about 22% using mesenchymal stem cells. This is a different kind of stem cell one gets from adult marrow or cord blood or some other source. But heart is really a major problem. We have high hope for islet cells, for insulin-producing cells. Perhaps the rejection, the immunological rejection phenomenon has to be worked at. But it is possible to generate large numbers of these animals that have pancreases that have human insulin-producing cells, and then get those and just isolate those. I mean, we can do that. I mean, it's not hard to isolate the islet cells and then just have large numbers of these cells. You know, in the liver you can have billions of human cells, even though the level may be only 10% or so sometimes. And in the pancreas, you know, you just keep doing more of the animals. So that's...
Yes, Dave.
Very interesting presentation. I'd like to ask what I guess is probably at this point a hypothetical question. If you're going to develop these cells for a particular individual who needs a transplant, how long is this going to take? Is it weeks? Is it months? I mean, you know, they're on waiting lists sometimes for years. What do you estimate, I guess, I would say?
The earliest we could do is 3 weeks. And we haven't yet looked to see we can facilitate this further, you know, speed it up. Between 1 and 2 months, you have a liver that is ready for transplant purpose. And pancreas and other things have already begun to show. Actually, liver takes a little longer to come on board for the purposes we are dealing with. But within 1 to 2 months, you could. But you know, we could have generic stuff just ready for this, or that could be used as a stopgap measure, or even take liver cells from some of the animals we have for detoxification of these patients, you know, in an extracorporeal manner, because they're now using pig livers to detoxify some of these patients. We could actually take human liver cells, and we're getting there in isolating them in pure form, and so detoxify these patients.
Dr. Zanjani, you use sheep as the host animal. Can you use other animals?
We think we can use pigs as well. I had a slide which I removed, which shows why sheep was so relevant to clinical medicine. You know, these animals, random breeders like us, you have 1 or 2 babies. A lot of things we've learned about human fetal physiology was learned in Cambridge, England, using the sheep as a model. The sheep also very nicely sometimes has models that duplicate human diseases. And there are other things about the sheep as well. We can't use animals that are too small. Rabbits and things cannot be used. But I think pig is probably the next possible species to use.
Yes, sir.
What's the importance of NIH support for this work as opposed to some private sector? So sometimes people ask, well, shouldn't this all be done in a company? Can you talk about that?
Well, we've been approached by companies, but they want us to do specific things. They have a particular set of stem cells they want us to use, and they want us to look at skin only, you know, and actually they prevent us from doing anything else. We've stayed away from that. NIH gives you the freedom to really put your scientific desires and know-how into practice. And you know, those limitations are in there. We are luckily being funded by NIH now, but of course we haven't gone for renewals yet, so we are worried about that. But NIH is so very important for these kinds of activities. First of all, they let you come with ideas to them, and therefore, and you know, your peers will decide whether what you're suggesting is a good idea or not, whereas companies tend to just limit you in what you want to do.
So, have you been funded throughout your entire scientific career by the NIH? Have you ever been funded also by the NSF? That's two different questions.
I've been very lucky. As soon as I got my PhD, I got my first grant, and I've had it ever since. Not that particular grant, but
How long?
it's almost 40 years.
40 years
Yes.
of funding from the federal government?
Yes, NIH. And I've never had an NSF funding. I've had private foundation fundings in some of the areas, especially in the in utero gene therapy they've been interested in.
All right, one last question for me. What will be the first time that this science you're talking about, theoretically, will be applied in something moving health forward in the United States? The first instance of concrete result.
You know, I know you all have heard that things like this happen in the next 5 to 10 years, clinical applications. But really, some of this has moved forward so rapidly that it could be much sooner than that. All one needs is funding to do this. I mean, right now, I think it is possible, for example, to do the detoxification studies in patients within the next 6 months because these livers are available for these type of studies. But to really do a clinical transplant, and the proof of principle studies have been done, so to do a clinical transplant, one has to just have the funding to do it. And I think it's really closer than further away. I don't want to give you a date.
One final question. Will you wait for the mic, please, sir? Wait for the mic, please, sir.
I'm a little concerned about the immune rejection. Is there any bystander rejection from other tissue? You said they will just cleanly reject the animal tissue, but is there any chance that you would have more widespread immune rejection? What are the side effects of this immune rejection?
Well, the only information I can give you is when we put the pig liver into the sheep. And there, the only thing we saw was the rejection of the pig cells in the liver. Now, in the general area, there are some deterioration that takes place, but and there were changes in the peripheral blood picture of this animal, but ultimately there was not any long-lasting side effect. And when the initial rapid rejection of the animal cells occurred, the liver began to regenerate and things just quieted down very nicely. So I don't have definitive answers for you except the observation I just mentioned.
Dr. Janjani? Oh, yes, yes. Just
The issue of chimeras have been fairly confusing and topical here in Congress in the past year or so, and a lot of people are concerned with studies that have come out with human cells going into the brains of rats and such. And what do you say to those folks that are A, doing that type of research or want to do that kind of research, and B, to those of us in Congress, or the people here that are representing members of Congress in terms of oversight in that particular area? Because it seems that there should be, or it can be argued that there should be some sort of strong oversight where some of the more ethical issues are raised.
I actually feel that a strong oversight on those particular areas are important. I wonder if the overall product of what we know and our consciousness depends not only on what we have in our system, but also our experiences. You wonder if a mouse that had a major portion of his or her system to be human neuronal or structural elements will get to have the same kind of experience we as a kid and so on develop. Or whether it would be compatible with the life of the animal, whether it can actually function to sustain that animal after birth. It's possible that those animals will die before birth. I would be careful. I think what Stanford had suggested, people at Stanford, that you begin looking at these animals soon after you do the transplants to see at what level they are and if they're getting to anything that would make them human. And this is an intuition on our part. I mean, how would we know? They can't talk to us. Then I think you should terminate and do the studies as you go along. I feel a little concerned about that too. That's why we have not as yet attempted to put the cells directly into the central nervous system to see if we can, we can actually concentrate by putting human stem cells in these animals for the central nervous system type stem cells. We've done that. It's the same cell that Weissman has reported. We haven't published it because you just don't know what to do with it yet. And we think that maybe if we put those cells into the central nervous system, we'll have a sheep that has a higher level of human neuronal or other structural cells within the system. But we feel a little uncomfortable about that.
When they say start playing golf and tennis, it's going to start time to worry about it, right?
Yes?
Thank you very much, Dr. Sanjani.
Well, thank you.
We want to thank the Joint Steering Committee for Public Policy for sponsoring this luncheon. I want to tell you, the last briefing in our series of this year will be on September 27th, a couple of weeks from now, and the subject is gray hair and skin cancer. I don't know how they're related, but we're going to find out. Gray hair and skin cancer. Thank you all very much.